This video explores complex biological phenomena for educational purposes and does not encourage dangerous acts. Deep breath. In the freezing, high-pressure depths of the abyssal plain, a massive cartilaginous silhouette materializes from the absolute darkness. Symbiosis microcephalus glides through the water column and propelled by slow, methodical undulations of the axial musculature. Belonging to Symbiosis microcephalus. The metabolic rate of Symbiosis microcephalus is perfectly calibrated for absolute energy conservation in an environment devoid of solar radiation. Yet, Symbiosis microcephalus does not navigate the abyss through visual perception. Attached directly to the corneal tissue of Symbiosis microcephalus is Omatocoita elongata. Omatocoita elongata is a specialized parasitic copepod that permanently anchors the feeding apparatus of Omatocoita elongata into the ocular flesh of Symbiosis microcephalus. The infiltration by Omatocoita elongata consumes the corneal mass, inducing severe lesions and near-total blindness in Symbiosis microcephalus. Symbiosis microcephalus possesses no biomechanical mechanism to dislodge Omatocoita elongata. Instead of succumbing to starvation, Symbiosis microcephalus relies entirely on the highly developed olfactory bulbs of Symbiosis microcephalus to detect decaying biological matter sinking from the photic zone. Establishes the brutal reality of abyssal ecology. In this nutrient-starved expanse, independent survival is often an impossibility. Biological entities must continuously exploit the physical architecture of neighboring organisms. Omatocoita elongata exploits the immense geographical range of Symbiosis microcephalus, utilizing Symbiosis microcephalus as a living biological transport vessel to filter the surrounding oceanic currents for reproduction. The blindness inflicted upon Symbiosis microcephalus shatters the illusion of peaceful ecological coexistence, revealing a system of forced biological taxation. Omatocoita elongata extracts physical anchorage and motility, while Symbiosis microcephalus endures permanent ocular degradation. This extreme dynamic demonstrates that deep ocean adaptation relies fundamentally on ruthless physical merging, setting a precedent for even more complex biochemical enslavements. Located deeper within the benthic topography, far below the pelagic zone, the abyssal plains fracture, giving way to immense geological ruptures. Here, hydrothermal vents violently expel superheated fluids saturated with hydrogen sulfide, heavy metals, and methane. This volatile chemical cocktail is fundamentally lethal to standard carbon-based marine biology. Yet, Thriving directly upon the scalding mineral chimneys is Riftia pachyptila. Riftia pachyptila constructs rigid, chitinous cylindrical tubes, anchoring the basal structures of Riftia pachyptila to the unstable basaltic crust. Riftia pachyptila rapidly generates dense, towering biological forests in an environment completely devoid of solar radiation. Riftia pachyptila presents a severe biomechanical paradox. Upon physical maturity, Riftia pachyptila completely sheds all digestive anatomy. Riftia pachyptila possesses no mouth, no stomach, and no intestinal tract. To secure energy within the lethal chemistry of the vent fields, Riftia pachyptila extends a highly vascularized branchial plume directly into the toxic hydrothermal gradient. The brilliant crimson coloration of the plume extending from Riftia pachyptila is not decorative. The coloration is generated by a highly specialized, complex, extracellular hemoglobin, saturating the circulatory system of Riftia pachyptila. This unique hemoglobin structure permits Riftia pachyptila to simultaneously bind oxygen from the ambient seawater and highly toxic hydrogen sulfide from the vent emissions. If a standard marine vertebrate absorbed this concentration of hydrogen sulfide, cellular respiration would instantaneously terminate. Riftia pachyptila, conversely, actively and deliberately harvests the lethal toxicity. The vascular network of Riftia pachyptila functions as an extreme biological pipeline, continuously pumping the volatile chemical mixture deep into the internal cavity of a deep-sea tube worm, Riftia pachyptila. Because Riftia pachyptila lacks a digestive system to process particulate matter, highly specialized internal organ called the trophosome. The trophosome isolates the captured toxic chemicals from the surrounding oceanic water, establishing the precise biochemical conditions necessary for the next phase of deep ocean survival, transitioning the narrative of the abyss from mere physical anchorage to profound internal exploitation. Within the trophosome of Riftia pachyptila, an extreme biological reality shatters the traditional ecological myth of harmonious mutualism. However, the internal mechanics of Riftia pachyptila represent an architecture of ruthless biochemical enslavement. The trophosome of Riftia pachyptila houses billions of individual Candidatus endoriftia Persephone bacteria. Candidatus endoriftia Persephone requires the exact volatile compounds pumped in by the branchial plume of Riftia pachyptila to survive. Once Candidatus endoriftia Persephone enters the trophosome, of Riftia pachyptila. Candidatus endoriftia Persephone utilizes the delivered hydrogen sulfide and oxygen to power chemosynthesis, effectively stripping chemical energy to fix carbon into organic molecules. Riftia pachyptila does not merely passively receive a share of these organic molecules. Instead, Riftia pachyptila aggressively harvests the metabolic byproducts of Candidatus endoriftia Persephone and continually digests populations of Candidatus endoriftia Persephone directly to sustain the massive growth rate of Riftia pachyptila. This interaction is an industrial-scale biological factory where Riftia pachyptila fully controls the vascular supply lines. Candidatus endoriftia Persephone functions merely as an internalized enzymatic engine, stripped of independent motility and subjected entirely to the metabolic demands of Riftia pachyptila. Without the specialized hemoglobin and rigid containment structure of Riftia pachyptila, Candidatus endoriftia Persephone would be dispersed and eradicated by the hostile vent fluids. Conversely, without the carbon-fixing capacity of Candidatus endoriftia Persephone, the digestive-free anatomy of Riftia pachyptila would undergo immediate cellular starvation. This mandatory biological merging forces immense evolutionary success and late, allowing these hijacked biological combinations to construct sprawling benthic ecosystems that fundamentally alter the surrounding geological structures. Ascending from the geological chaos of the hydrothermal vents into the absolute sensory void of the bathypelagic zone, the necessity for biological exploitation takes on a predatory architecture. Hovering motionless within the freezing water column is Melanocetus johnsonii, the highly textured, light-absorbing epidermis of Melanocetus johnsonii. Renders the physical form of Melanocetus johnsonii entirely invisible against the abyss. Extending from the dorsal cranium of Melanocetus johnsonii is a highly modified, articulated appendage terminating in a fleshy, within the crushing darkness, the ESCA emits a precise blue-green photon emission. However, the cellular machinery of Melanocetus johnsonii completely lacks the biological capacity to generate this bioluminescence. The photon emission is forcibly outsourced. The esca of Melanocetus johnsonii is not a simple anatomical lure. The esca is a highly specialized bacterial containment and cultivation chamber. Housed within the intricate tubular matrix of the esca are dense, localized populations of Vibrio fischeri. Vibrio fischeri is a free-living, bioluminescent bacterium. From the surrounding oceanic currents during the early developmental stages of Melanocetus johnsonii. Once Vibrio fischeri is trapped within the esca, Melanocetus johnsonii assumes absolute biochemical control over the internal environment. Melanocetus johnsonii dictates the reproductive rate and metabolic output of Vibrio fischeri by restricting or flooding the internal capillary network of the esca with vascular oxygen. This precise vascular manipulation allows Melanocetus johnsonii to dictate the exact intensity of the photon emission generated by Vibrio fischeri. The metabolic byproduct of Vibrio fischeri to deceive visually oriented biological targets. Vibrio fischeri receives a stable, nutrient-dense environment, but strictly at the biomechanical direction of Melanocetus johnsonii. Permanently incorporate and manipulate the cellular functions of Vibrio fischeri. To construct a fatal optical illusion. Resting upon the benthic sediment is the colossal, decaying skeletal structure of Balanoptera musculus. The calcified remains of Balanoptera musculus represent a massive, temporary, localized energy reservoir within the nutrient-deprived abyss. Colonizing the exposed osseous tissue of Balanoptera musculus is a dense biological carpet of Osedax muciflorus. Osedax muciflorus presents yet another extreme anatomical deviation driven by the necessity of biological exploitation and interaction operating far outside the confines of traditional parasitism or mutualism. Similar to the anatomy of Riftia pachyptila, Osedax muciflorus entirely lack was a gastric cavity and oral aperture. Osedax muciflorus cannot physically consume the lipid-rich marrow trapped within the bones of Balanoptera musculus. To extract this sequestered energy, Osedax muciflorus secretes a highly concentrated biological acid. However, The acid secretion generated by Osedax muciflorus only grants physical access. To actually metabolize the complex lipids, Osedax muciflorus relies on a massive, with internalized population of Oceanospirillales bacteria, housed within the root system of Osedax muciflorus. Osedax muciflorus forcibly isolates Oceanospirillales from the benthic environment. Oceanospirillales chemically degrade the exposed marrowlipids, synthesizing organic compounds. That Osedax muciflorus continuously absorbs across the cellular membranes of Osedax muciflorus. This interaction is an aggressive, mechanized extraction protocol. Osedax muciflorus. Furthermore, the physical structure of Osedax muciflorus visible upon the bone strictly comprises the female physiology. Microscopic male specimens of Osedax muciflorus exist permanently trapped within the gelatinous casing of the female Osedax muciflorus, entirely dependent on the energy extracted by the captive Oceanospirillales. This multi-layered exploitation showcases the extreme limits of biological reliance, hijacking microscopic machinery to unlock energy reserves that would otherwise remain permanently inaccessible. The brutal reality of the deep ocean shatters the terrestrial illusion of peaceful ecological mutualism. From the freezing pelagic zones to the boiling hydrothermal vents, but by the mandatory hijacking of neighboring biological architecture. Symbiosis microcephalus surrenders ocular function to transport the reproductive cycle of Ommatocoita elongata. Riftia pachyptula constructs toxic biochemical prisons to perpetually harvest. The metabolic carbon output of Candidatus endoriftia persephone Melanocetus johnsonii ruthlessly manipulates the vascular oxygen supply of the esca to weaponize the photon emission generated by Vibrio fiscuri. These biological relationships are not harmonious collaborations. These biological relationships are aggressive, biomechanical optimizations. The deep sea is a nutrient-starved void that demands absolute metabolic efficiency. To achieve this efficiency, biological entities must permanently cross the boundaries. The organisms that dominate the abyssal plains are the organisms that successfully enslave the specialized microscopic engines of the deep, building thriving biological oases through calculated exploitation rather than passive cooperation. The true architects of the abyss are the invisible biomechanical connections that bind predator, parasite, and symbiote into singular, highly functioning survival machines. The deep ocean requires biology to consume biology from the inside out. To continue exploring the hyper-realistic mechanisms of evolutionary biology, and the hidden, unyielding architecture of the natural world. And please leave a like on this video and subscribe to the channel